Yeah, now
I think it is recording, sir. Yeah.
yeah, yeah,
It is
it is
being
not
recorded.
visible. Must. Okay. Okay. Very good afternoon to all who joined in this online program as a part of regular monthly talks. Before the talk starts, I will give brief introduction about today's speaker. This is Mr. Sarvan Kumar Parepalli. He did his master's in biochemistry from Andhra University with distinction. And then he qualified for All India NET in CSIRI and worked in CSRI IICT lab for 2 years. Then he joined IHR in 2016 under the guidance of Dr. Kamala Jayanti as SRF and followed by in a national fellow project. He has vast experience in handling GCEAD and GCMS and published 26 papers in peer-reviewed journals, and he is associated in filing 2 patents, and he registered PhD in the year 2018, and he'll be submitting within a week his thesis. Then he attended 5 international seminars and gave oral presentations as well as some poster presentations also. And he is part of the 2 big projects. One is National Fellow Project and another is presently he's working under BBT, Northeastern Region Chemical Ecology Project. And he is already selected as scientist in one of the labs in California in the United States of America. Anytime he can go and join there. He himself can give some details about his future recruitment and his job in ES. With this brief introduction, I request Shravan Kumar to give a talk on the topic Deciphering the role of gut enzymes, endosymbionts, and semiochemicals in greater wax moth, that is Galleria mellonella. Over to Shravan Kumar.
Yeah, thank you so much for the kind words. So far, nobody has introduced me like this. This is the first time I should say this.
Yeah. When you go to ES, we request you to be a speaker for our international seminar, which will be held in 2013. Please keep this in mind. Sir. Not only yes, wherever you are, definitely you will be in yes in the coming 6 months. You also give some of the staff wherever in whichever university you are going to work. Please go ahead.
My pleasure. It will be my pleasure, sir. I will definitely do that.
Yeah, please go ahead.
Okay. Yeah, I'll be sharing sir now screen. Okay, so can you see, sir, my screen? Is it visible, sir?
Yeah, it's visible. Clear.
Yeah.
Okay. Fine. So, very warm good afternoon to everybody. And my name is Shravan Kumar. I'm working as a research associate in Indian Institute of Horticulture Research under the guidance of Dr. Kamala Jayanti. And this talk is related to my PhD work, and it's a title called Deciphering the Role of Gut Enzymes, Endosymbionts, and Semiochemicals in Greater Wax Moth, Galleria mellonella. So to give a brief introduction about it, as you all know, honeybees collectively take a lot of efforts to collect the honeycomb, and but poor fellow they doesn't know whether even this honeycombs also can be destroyed by a pest. So that pest is called Galleria, that is called greater wax moth. So these pests, it attacks this honeycombs and it completely destroys it, and it makes good-looking honeycomb wax into this kind of degraded one. So coming to an introduction for this pest, this is Galleria wax moth is also called as greater wax moth. It's belonging to the family Pyralidae and order Lepidoptera. It is the first reported pest in Asia, but now it is widespread and ubiquitous. And it is a severe field-based honeybee. It's a pest of severe-based field-based honeybee colonies, also the stored honeycombs. And it is also almost causing 60 to 70% of loss in economic in agriculture industries, and even in the field, even in the fields where this honeycombs or beehives are kept. So usually empty honeycombs, the honeycomb which is not properly guarded by honeybees or which is left attenuated, attracts this wax moth and they infect it. So usually this honeycombs, the wax moth life starts with the eggs, as everybody knows. So eggs it hatches to first instar larvae, and the larva instar tail raises to 6 to 7 instars. From there, it will develop into pupae and pupation to adults. So coming to the economical crisis, what had developed with this wax moth infestation in India, actually, this is the data of very old, some of the literature paper we have used a source. But recently there is no report with the damage of wax moth, because I think nobody is working on it and nobody is giving the reports like so and so economic loss has been observed because of the wax moth. But as of now, with this available data, it was reported as 50 to 100% of economic loss was observed in 1983. And which was reported by the Kapil and Shihak, and not in India, overall in the world, like USA almost faced $3 to $4 million loss in 1976, even in the Florida and even in the Iran with 38% of economic loss. And even in the Pakistan, the side by country, they have facing the problem with the wax moth. So what are the existing methods which were available to eradicate, to control this wax moth? So these are some basic headings which I am giving, to which earlier the methods are developed to control this wax. Out of this, like chemical methods are there, cultural practices are there, some of the biological control methods, and SITs, and moreover maintaining the good hand sanitization, using of trichogama as a species, using of beetles like Brachyonhibitor and Apantellus Galleria, and raising the temperature and cooling down the temperature. All those kind of activities were performed to control this wax moth. But now recently, some of the farmers in USA, they have found interesting traps, which is very feasible and very easy to make. You can see here, they have just using the glass, this plastic bottle, they have made a hole and they are putting some water into that with a cup of sugar, a little of hot water, and a cup of vinegar, cider vinegar or any other vinegars. And after that, they are putting the banana leaf into that and they are allowing it to get ferment. So once the fermentation starts, the cues from this, it's attracting this wax moth, what they have reporting. So this may be a very easy method which they are applying with their own knowledge. So what are the present scenario? Can a farmer or can any of us use this beekeeping idea to get profitable amount, or they can use this as their source of income? Yes, you can do it, but the only thing, the problem here is if you can maintain a good sanitation, good practices. Without this is very difficult, because once you doesn't look into that or doesn't put your heart of making the beekeeping, honeybee production and all, this wax moth is going to affect it and is going to completely lose your money and your income also. So let us quickly see the nature of damage of this wax moth. So how it damages in the field. So usually the wax moth gets mated at the beside the tree wherever this honeycombs or beehives are kept, and they get gravid females, and they become mated and they become gravid females. And once there is a loads of eggs present in the females, they try to infect the honeycomb in the night time, because this is a nocturnal pest. And this is very interesting feature of this insect, because the honeycombs are very active in the daytime, and after that, after all the worker bees which goes and takes all the pollen and honeycomb, they come in the evening and they'll relax and they are not so active. So taking this as a crucial time, this insect invades, affects and attacks this honeycomb, and they locate a particular site wherever they feel better, and they try to see which is not affected by the bees, and they try to lay the eggs into it. And once the eggs are laid into a particular honeycomb, wherever little cracks and crevices are there, from there within a week the eggs develop into larva, and larva as soon as it emerges, as soon as it hatches, it starts feeding on the honeycombs like anything. It is a very vigorous feeders, and it completely finishes the wax moth within no time. And after that, it also makes a galeriasis, forming a complete web-like structure on the brood. And because of that, the newly formed whatever honey bee pupae are there, They are failed to emerge into as adults, and they're all called Galleriasis. From there again, it becomes into a pupation at the corners of the beehives, and after that it develops into adult, and this continues the life cycle. So with this all knowledge, next we framed some of the objectives to focus on this work and to control this wax moth. So at first, as a chemical ecology lab, we wanted to understand the role of potent semiochemicals, which is involved in the behavior of both larvae and adult moths. Next, to know why is this wax moth has taken primary host like honeycomb, because it is made up of wax and it is difficult to digest this wax as even for humans or even for another organism. How this tiny organism is able to degrade it. So to decipher these reasons also, we wanted to see this gut endosymbionts and also the enzymes which is associated with the degradation for wax moth. So this is a recent literature review I wanted to share with you guys. So recently in 2022, a paper has been published from in the Nature Communications, because it's already known fact this wax moth is able to degrade the polythene, and it's a very interesting feature for this, because you all know this research was started in 2017. Accidentally in the Germany, people were using this before taking this as a wax degrading or polythene degrading organism. This insects were used as a model organism or model for human pathogenesis, or even for in vivo testing, and even our Uma ma'am also uses in her lab for nematode testing and all. So in this in this kind of work, they were keeping these larvae into polythene, and they have observed the polythene has become holes inside it, and the larvae were escaped. So this was a very interesting feature what they have observed, and from there the research started, and they have published it. They almost took 4 years, right? When I have registered this work in for my PhD work, but I didn't select it for degradation of polythene, but I degraded for the host that is called honeycomb. So they have published recently saying that the saliva of this waxworm larva contains some of the enzymes or some of those microbes which are able to degrade this polythene. So they have very nicely identified some of the genes also responsible for that. So whoever interested, you can read this paper. Apart from that, in 2021, one paper was published which knowing that what are the microbes which is available in the wax moth and whether these are responsible for polythene degradation. So with that, they have published some of the microbes which is essential in degrading this polythene contents, and they have also identified some of the microbes. But all of this literature says only this wax moth is useful for model organisms or useful for polythene degradation. But nobody has till now worked on semiochemical aspect or even the olfaction aspect and the original how it is able to degrade the bee wax. So that is most important to identify what is inside happening with this insect. So with this knowledge, we have moved on to identify these unattended questions and unexplored one. So basically, everybody knows the insects are attracted towards this host with the use of semiochemicals. So semiochemicals play a crucial role in any insect to find its host and even for the oviposition. So whether these cues can be collected, whether these cues can be utilized to attract these forms. So with this knowledge, we have developed our methodology of collecting this honeycombs as like beehives, and we have also developed culture in our lab with the help of GKVK. We could took initially the larvae from them, and we could establish the culture in our lab. So we try to take the headspace volatiles of beehives, which is intact and which is present with the bees, and the one which is not having any bees, which is BW, which is beehives without bees, and the other one which is beehives with conspecific, which is already infected with the wax moth larvae. So the reason for collecting these 3 types of volatiles is because in general, in nature, how's this, even the wax moth, if it infects, it has to infect on a particular bee honeycomb. So it may contains a lot of bees inside it. So initially to know whether realistically, whether this honeycomb, whether the wax moth is able to attract to those which is intactly developed with the bees, or it is only tricking only with the wax. So to answer all these questions, we have taken the volatiles of all these 3 treatments, through air internment method, which is customized and which is like already the methodology already published in several papers. And we have collected these volatiles in the night time and till evening 7 o'clock to morning 7. And we have eluted and we have performed some behavioral Y-tube olfactometer bioassays. So we have performed behavioral bioassays with both of the larvae and adults. We have used gravid female adults here, and the 3 to 4th instar, which is aggressive feeders, we have used to know whether these are attracted towards the collected volatile cues or not. So first we have used for a larva and we have found, okay, we'll go for the results next. And after this behavioral bioassays, we have performed electrophysiology studies also with the antenna of this wax moth to identify the cues which is responsible for attraction towards this host. So coming to the results, first we are talking about the larva here. So we have taken the results and we have integrated this analysis, and we have checked that the larva showed non-significant behavior with any of the cues, because we thought maybe the larva is not responsible for detecting the cues, but in the field or in the lab, whenever this honeycombs directly, the comb piece of comb has kept, it is moving towards it. But the reason of here to perform the Y-tube assays, because we wanted to see whether the larva could also has some senses or can detect some specific cues or not. But unfortunately, we couldn't able to get any significant data from this analysis. And even to perform the EAG studies with larva were difficult because of the continuous movement of this larva when we are keeping into a this manipulator, the electrode probe. So avoiding this, then we have moved on to know the results of adult moths. So in adult moths, we have found very interesting data. Here we have find that female wax moth showed a very significant response to the volatiles which is collected from BB, that is beehives with bees, and also the beehives without bees. But it not preferred the beehives with conspecific, showing that the one which is already infected with the larva were not preferred by the wax moth. So this was the results in the case of single choice. Next, we have performed the dual choice also with the giving the combination and making the insect to choose between the two treatments, which we have observed. Insignificant response was observed when we have given the choice to select BW and BB. But significant response was observed when the choice was made between BW and BC, where the insect was almost moved towards BW, which is beehives without bees, but it didn't prefer the conspecific cues collected one. The same case with the BB and BC also, it showed significant behavior. So overall, with these results, we have conducted an interaction index, and we have identified here is the wax moth is not preferring the volatile cues collected from BC, but it is showing highly attractive towards BW and BB. So surprisingly, we would like to know like, how is this happening? Because a wax moth should avoid the volatiles collected from BB because it's already contained honeybees along with the beehives. But surprising to us, it didn't show any non-significant behavior. It showed attraction. Later on, we moved to doing of EAG study also, and here also the trend was continued, and we have seen the response was good for BB, BW, but a little less with BC. However, there was no significant significant result shown between these treatments. But when you compared with the control solvent, the significant behavior was observed. So further, with this idea, we wanted to study, okay, so now we have got an idea to know whether the volatiles of BW and BB shown attraction for wax moth. Can we identify what are the cues responsible for it? So here is this GCAD results which performed with the females, gravid females in the evening time with the headspace volatiles of BB. And we could identify almost 13 chemical cues consistently gave a good response. And these are the compounds which we identified from the BB, which is lots of these chemicals were acids and aldehydes, and some of them were sesquiterpenes. So interestingly, we have identified here a chemical called octyl acetate, which was reported as a alarm pheromone in BB, which is also called as a danger pheromone, which is produced by the honeybees whenever an attack by wax moth is observed. So moving on for the EAD performance with the volatiles of BW, here also we have identified a very consistent response to 14 chemicals. And these are the chemicals which all contains most of the acids and esters. But along with the esters, interestingly, we have found a chemical called phenol and osmin. These are the terpenes, monoterpenes, and ethyl benzoate and indole, and also alpha copene. So these chemicals were also reported as some of the attractants in some lepidopterans. But here in our study, we have to identify whether it's really attractant or not by performing a synthetic bioassays, by purchasing these chemicals and performing Y2 assays. So before doing that, we also compared the GCMS profile of all these three collected volatiles. And here we have identified a lot of the compounds were sharing between BW and BC, and there are a little higher amount of esters and acids were more in BB. So that was really interesting one, which we have taken into account, and we have moved on for doing synthetic bioassays. So synthetic bioassays with all the individual EAD active compounds, which we have identified in electrophysiological studies. We have purchased some of the chemicals which were available in the market, and we performed a single choice assays. And here you can see the chemicals which is marked with the yellow color showed a highest good response, I mean almost significant response with the gravid female wax moth. So these again, the chemicals which were identified from BB and the chemicals which were identified from BW were separated out, and a synthetic blend was made with the two different synthetic blends were made with the chemicals which were isolated and identified on particular treatment. So here also interesting results we have identified. We have found that the moths preferred the synthetic blend of BB, almost 70% of making choice, and showed significant response. Same with the case with BW, which showed 75% of choice percentage. But when we gave the dual choice assay, between the BW and BW synthetic blend, between the BW and BB synthetic blend and BW, there was non-significant response. So this is giving an idea that what are the compounds we have identified when we have given a dual choice assay, and we are allowed to insect to choose between the synthetic blend made of the EAD active components of that particular treatment, along with the natural volatile, which is collected for the particular treatment, there was a significant response. So that means whatever the volatiles cues you have identified has an attraction property. So with this knowledge, we again performed ovipositional bioassays. We have taken a small box and we have released 2 filter papers, and we have released some 5 insects, and we have released this individual cues inside it, and we have counted the eggs. So interestingly, we have found that the oviposition index, when we calculated based on the number of eggs which is laid on a particular treatment, the wax moth showed highest number of egg laying in the volatiles which was collected from the BW, and interestingly not from the BB and BC. So far we have seen that the volatiles collected from BB and BW shown attraction when we have performed a behavioral bioassays and we have performed EAG studies. But when you are literally giving ovipositional studies, ovipositional assays, and you're literally giving a platform to insect to go and land on a particular arena, it is not preferring the one the volatile which is already it shown attraction. So this makes this little interesting thing, and it made us to see into some different literatures, like what is happening inside it with the behavior of this wax moth. Apart from this, we also tested with the synthetic blends which we have made here, and synthetic blends also, the blend which is made with the bee hives without bees showed good number of eggs compared to BB. So this was also a trend which is we are now checking, and we have seen with the earlier results. And down here we can see with the individual chemicals, whatever we have identified from the EAD studies, individual chemicals also we have performed oviposition bioassays, and the chemicals like ethyl isovalerate, phenol, heptanoic acids, nonanol and nonanic acids, undecane, ethyl benzoate, and cariflin showed highest number of eggs when you have done the ovipositional assays. But the chemical like octyl acid, which is already have just now told, like as it acts as a alarm pheromone for bees, didn't show any egg response, very few, and even for the limonene, osmin, and alloosmin. So with these results, we tried us to see the literature, like what is exactly happening with the insects. Other than that, it made us to think about whether not only the antenna, whether the insects can sense the odors from any other organelles. That was a question. So based on this question which we have made to ourselves, we have found some literature, and the literature says, apart from the antennal thing, for an insect to see its oviposition, to locate its oviposition, to lay eggs, and to particularly select any host, not only the antenna, it also requires some other organelle, some other senses, which is highly important in, especially in lepidopterans. So there are some papers which is reported in Nature saying that the antenna, the senses of tarsus, the forelimbs, has some chemical, some chemical chemosensillus, which has the ability to detect the specific cues when insect go and land on a particular host. Same with the case of any other beetles. Here also we can find they are reporting, like, if the insect is locating any particular host and it is landing on it, the first body parts which lands on it is tarsus. So when the tarsus is touching towards any particular host, the surface of it gives some of the senses which was received by the receptors of this tarsus, and thereby it goes into a contact chemoreception, and from there the motor sensory neurosystem activates the brain of insect, and it makes a choice and it decides whether to oviposit at a particular site or not. So this was very interesting observations which we have observed, and with this idea we later on performed, based on these results, we have performed EAG studies and some bioassays. And here we have identified what we have observed here is, in our case, wax moth also using two different sensory modalities to evaluate the suitability of potential oviposition sites. So how we can conclude this is, we have dissected, we have excised the tarsus of this wax moth, and we have did a study called electrotarsogram, where we have used a filter paper and we have put this volatile cues inside into it, and we have slowly touched the sensillus of this tarsus, and we have recorded the observation. As you see the millivolts response, though the response was similar to BB and BW, but here also it didn't prefer to BC and even the control filter paper and the solvent. So this was a little interesting data which we have got to move further on to develop a neuronal model hypothesis, where we have developed, we have considering the signal integration from the olfactory and contact chemoreception system in the wax moth. We have developed a possible neuronal models. So here, when an insect is, when a wax moth is sensing the volatiles of BB, so the olfaction system in it activates, and it makes the insect to move towards the beehives. But whenever it is landing upon a particular beehive, the contact chemoreception, that is the tarsus, it is giving a negative signals to the motor sensory neuron, where the insect is able to know, like, it is a danger signals is coming from the beehive, means the beehives, because of the presence of bees. So now that is restraining the oviposition. But when insect is sensing the volatiles or the cues from BW, means the beehives without bees, so the olfaction is instigating the insect to move or fly towards that particular beehives. And upon landing, the contact chemoreception on the tarsus is giving a positive signals to the motor sensory system, and which is stimulating the oviposition here. But this is what's happening with the BB and BW. But in case of BC, when the BC is when the insect senses the BC, it is seeing that the olfaction is, or even though triggering it towards a flight towards a particular beehive, the contact chemoreception system is giving a negative signal, so that it is also restraining the oviposition. So this is a very interesting data. What we have identified is, the insect uses its senses even from the antenna and even from the tarsus of its body part to locate a particular oviposition site and to lay the eggs on it. Because as you know, for insect to oviposit and to get into that particular thing, it requires more observations, and it is a very costly procedure for that, and it cannot simply give away like that. So it has to think in a different directions, and it has to be different parameters to make sure that the offspring which it develops should be perfect and well grown. So this data clearly supports the neuronal model, where the central integration signals from olfactory and contact system is mediating the risk detection by female wax moths. So this is the paper which we recently published in scientific reports, and it is recently accepted. And with all this data, we have moved on the erecting of delta traps to trap the mellonella. But unfortunately, maybe there is something which we have to work on more if you trap the insects in the field, because whatever the results which we are getting in the lab, it is not enough for trap the insect in the field. So the standardization of this process is happening in the underway. So now the story is not end here. So now we have observed, like, okay, we have identified some of the cues and we have prepared a blend which it attracts it. But to know what is happening in the inside of the insect antennae, how the olfaction perception is happening, and what is the procedure, the mechanism behind the olfaction in an insect. So usually, very brief presentation which I want to share here is, so once the antenna of an insect receives the signals from any of the host cues, so the sensilla which is present on the antenna receives the ligand, and the proteins, the specialized proteins which is present on the antenna is called odorant binding proteins, which receives this ligands, and there will be a binding to of both of it. And once this binding happens, it triggers and activates the olfaction receptors inside it. And when the olfaction receptors is activated, thereby biochemical effect and the behavioral mechanisms all starts. So with this knowledge of literature, we have tried of identifying what are the chemosensory genes probably available in greater wax moth antenna. And this also we recently published in Current Science, and we have identified almost 24 of odorant binding proteins, 7 antennal binding proteins, 6 pheromone binding proteins, 62 odorant receptors, 4 ionotropic receptors, and 2 sensory neuron membrane proteins are identified and confirmed in the wax moth. So this confirmation was done with the help of software Mega, and we have developed a phylogenetic tree with the maximum likelihood tree, and the tree clearly represents whatever the odorant binding proteins which we have identified has a maximum sharing capacity with this other insects of this belonging to the same family called Bombyx mori, Mandosixta, and Helicoverpa alzimera, both in the case of odorant binding proteins and antennal binding proteins. And even for the ORs, the odorant receptors, also what we have got in this data is also have a maximum share with the other Lepidopterans, Bombyx mori, Sexta, and Helicoverpa, even with the case of sensory neuron memory proteins. So now we have got the odorant binding proteins number, and even we have confirmed it sequences. So what we can do with this kind of data? So there is a concept called 3D modeling structure and molecular ligand docking, which is very booming nowadays, and even the pharma industries do this protein docking before releasing of any drug into the market. So taking this as a knowledge and this as an idea, we wanted to study what are the sequences we have identified, whether the sequences we can develop a protein model with that, and with the protein 3D modeling, can we dock with the ligands, that is the EAD active compounds which we have identified in our studies. So here is a free software available in Google called Swiss model, Swiss docking. With the help of the Swiss modeling, you can develop the sequences what you have got, and you can develop a 3D modeling structure of each sequence, and you can dock with the individual EAD compounds, which is a ligand. So these are some of the structures, the 3D modeling structures of the odorant binding proteins, which we have isolated in this study. And these are the ligand structure, which is already available in the Google. So what we have to do here is we have to select any particular odorant binding protein and a particular ligand to which you want to test. And this we have to dock in a Swiss docking system. So here you can see, once you submit this the details of this sequences, I mean the protein data bank along with the ligand details, here you will find the values, the full fitness values, and the delta energy capacity of your compound, that is your ligand binding to a particular OBP. So this results gives you an idea whether whatever the compounds you have identified has a greater affinity towards a particular protein sequence. So this is so far it's a time-taking process, and we already have finished some of the docking studies, and later we are like underway of doing the others also. So this is how we are doing and working on this, and this recently we published in a Current Science paper. So to look into the summary of this first objective, we have identified that the wax moths are preferring the BW and BB, which is beehives without bees and beehives with bees volatiles, but they are not preferring to the volatiles which is collected from conspecifics. So the volatile showed, the wax moth showed attraction towards the synthetic blends made of BW and BB in laboratory. Still, the standardization of the blend in the field is needed and it is underway. And if, when it comes to oviposition, wax moth interestingly laid more eggs in BW rather than BB, so which gives us an idea that wax moth integrates the signals from olfactory and both contact chemoreception, that is tarsus, to evaluate the risk in oviposition site selection. And now, whatever the identified odorant binding proteins, we developed a 3D modeling structure from that, and we have did a molecular docking with the ligands molecules available. Further, these proteins which we have identified and which we have docked can be knocked genetically using molecular tools like CRISPR-Cas9 to control this pest. So this is for first objective. Later on, we wanted to see what are the enzymes which is responsible for this bee wax degradation. So to know this, again, thanks to the literature, we again need not to see the composition of the beeswax, whatever, to identify what is there inside. There are a lot of papers which is showing the beeswax contains 75% of esters and 15% of fatty acids and other 10% of hydrocarbons and alcohols. So keeping in this mind, we wanted to see whether the gut which we have extracted from a larva has the ability to degrade this esters and fatty acids of this beeswax. So keeping in this mind, we have performed the enzymatic assays and we did esterase activity and fatty acids. So that is lipase activity. We performed this assay with the known protocols and used substrate called p-nitrophenol acetate and para-nitrophenol palmitate. We did ammonium sulfate precipitation with 40%, 80% of saturation. And once the saturations of the component of the proteins happened, we further performed Cephadix G filtration, and there each filtrate we have performed specific activity, enzyme activity, the fraction. A total of 25 fractionations were prepared, and out of that, sixth or seventh fractionation was showed as having a high enzymatic activity. So these are the results. For the lipases, we have seen the crude enzyme, for the crude enzyme, which was directly extracted, showed 2.3 protein concentration, where the total activity is 0.03, and the specific activity is also less. But as soon as you purified this protein with the help of Cephadix G100, the specific activity increased to 1.63, that is almost 10 folds high. So that gives an idea that whatever the purification we are doing it has the lipases activity, and the gut enzyme has the enzyme which degrades the lipases component. And with the same case with the esterase also, when we keep on filtering it, the Cephadix G-del purified component gives us specific activities almost 11.23, which was a very good purification. And this also gives an idea that what are the gut extracts enzyme we have taken out. It has also the ability to degrade the esterase component. So next we have to see, we wanted to check whether what is happening if you are increasing the temperature and decreasing the temperature, or if you're increasing the pH. That means the biochemical characterization with the different possibilities. So here we have found the lipases activity and even the esterase activity shown almost higher at the pH of 7 to 8, both with the lipase and esterase. And in comes to temperature, the activity was more observed at 37 to 40 degrees centigrade, both with the case of lipases and esterases. So here, whatever the purified enzyme we have taken, we have performed a native page for that, and we have got a good band showing that the esterase and lipases activity was observed in the particular extract. So here's the summary for this. We are successfully established and isolated and purified the specific crucial gut enzymes, namely esterase and lipases, from the larval guts of mellonella. And this study could help in synthesis of some of this enzyme inhibitors to control this insect, and thereby as a control of as a method in a pest management strategy. So this is also paper which we recently published in Pest Management in Horticulture Ecosystem, the APME journal, and this is for second objective. So coming to the third objective, which is also similar to that, but here it requires the extraction and identification of the gut endosymbionts, which is the microbes which is associated with this wax moth. So as we all know, microbes are very key, playing a very key role in any of the organism, whether it comes to metabolism, whether it comes to degradation or digestion or anything. So some of these insects in which the microbes plays a very important role in this digestion of a complex polymers, and such as cellulose and xylem, pectin, starch, and here in our case wax. So this study is to identify the bacterial species from the guts of greater wax moth, which degrades the base wax. So to perform this, which we have taken MSM media, that is minimal salt media, with the composition which is labeled here, and this we again inoculated with the 0.2% of beeswax as a sole source of carbon source. This beeswax, which is a commercial grade, we have procured it and we have added into the particular media, and we have incubated for 35 degrees centigrade for 7 days, and we have checked the growth of the bacteria. And every 4th or 3rd day we took the 2 ml of this culture, and we have again put it into another media freshly to avoid the overgrowth of this microbe. So after doing this, after 7 days of this growth, we have taken 1 ml of this culture, and we have put it in a spread plate agar, and we have for the isolation of the pure colonies. So again, this microbes were also enriched on a particular agar plates, which was enriched again with the wax, beeswax as a carbon source. So with this idea, we have again isolated some 13 pure isolates based on these characteristics. So whatever the isolates we have identified, we did a biochemical characteristics of all these isolates, and we have identified here is almost all the organisms showed non-motility except G7 and G11. And almost all the bacteria showed positive towards methyl red vasopressor test, apart from G7 and G11 and G13. And most of the organisms showed negative reaction with catalase reactivity and citrate utilization test, except G7. So again, if we also check the colony characteristics of this 13 pure isolates, and we have found here is out of all 13, maximum all the surface of the bacteria is smooth, and the shape was circular for most of the species except G7, which was rod, G10, and G11 and G13. The margin was entire for all the bacteria except G11, and the color was a different form. Some are the grayish, some were pale white, some were again white, cream white, and dull gray. And the elevation was again raised for all the organisms except G11, that was ambonate. So then we have performed the gram reactivity for each isolate, and we have identified all the isolates which we have identified here showed the positive gram reactivity with the diplococci as a structure, and except G10, GM10 and GM11, which is rod, and even the GM13 also. And we have also seen the oxygen tolerance, and obviously they all were facultative anaerobic in condition. So later on, we have moved on taking all the individual bacteria isolates and putting into a again MSM media enriched with the beeswax, the 0.2%. And this we have again raised, incubated for 7 days, and then we have checked the OD values of all particular isolates. And interestingly, we have found out of all 13 microbial isolates, the isolates called GM1, GM11, and GM13 showed the highest OD value, resembling the highest growth of the bacteria. So based on these results, we again wanted to confirm visual test with the spirit blue agar test, where the spirit blue agar test gives us an idea like whether your microbes has the ability to degrade whatever the lipase content of substrate is there or not. So we have used beeswax as a substrate, and also along with that a paraffin works as a control, and we have taken this into a plates, agar plates, and we have inoculated this 13 isolates. And again, as a trend continues, we have found GM1, GM11, and GM13 showed the very good growth on this spirit blue agar, and they tend to change the color even as soon as the growth is observed. So the blue color is changes to transparent yellow, showed that there is an activity of the degradation of the wax content with the help with these organisms. So further, we moved on to identify these organisms, and here we have found GM1 was called as Enterococcus cassiflavus, GM11 was Bacillus cereus, and GM13 was Lysonobacillus. And we have also submitted into NCBI to get the accession number of each individual organisms. Next, we have also performed the enzyme assays with the isolated 3 bacterial isolates. And interestingly, among these 3, GM11, which is Bacillus cereus, showed highest lipase activity and also the esterase activity, which giving an idea that among all the 3 isolates, Bacillus cereus is playing a very crucial role in probably the digestion of this wax component for the wax moth. So as a conclusion, we have isolated almost 13 pure isolates from the wax of the guts of this beehive, wax moth. And out of 13, 3 were isolated, and based on performance, we have identified, and we assume this as a crucial in wax degrading capacity. And further, the mechanism of this degradation and biochemical pathways should be explored much to know more insight into this study. So further, at the last, we have also moved for doing some metagenomic studies where we have want to know what is the unculturable bacteria which is present in this wax moth larvae. So interestingly, what we have taken here is we took larva which is fed on artificial media, and also we took the larva which was fed on the natural medium. So interestingly, when we did the metagenomic studies, we found almost 75% of Proteobacteria as a phylum in larva which is fed on artificial media, but the larva which was fed on natural media was high compared to the artificial media, which is 82%. So coming to the species level, we have identified unclassified Colobacteriaceae family for 24 to 24.5% in artificial media fed larva, but it was almost 29% in natural media, which was organism of Pseudomonas viridiflava. So this Pseudomonas viridiflava, which have identified here, is it's almost reported as some of the highly hydrocarbon degrading bacteria, and also the polythene degrading bacteria in contaminated, oil contaminated areas, and even for the one which the places where the polythenes has been, placed as a garbage and all. So this Pseudomonas viridiflava is also found in natural media, indicating that whatever the results we have found is confirmed, and it is showing the possibility of, this moth, this wax. The wax moth larva has the ability to degrade our honeycombs substances because it also contains the wax inside it. So this is a chrono charts for the comparative results for the larva which is fed on artificial media and larva which was fed on natural media. Here you can clearly see 22% of viridiflava which is seen in artificial media sample, whereas 29% it is seen in natural media fed sample. So with this, the anticipated outcome of the study could be, we can use this study to decipher the reasons of greater wax moth selecting honeycombs as a nutritional source, and what was the mechanism involved in it. We can decipher it. And obviously this information will be valuable for developing novel behavior-based control strategies. So with this study, I would like to acknowledge very few people and very important people also, my mentor and my guide, Dr. Kamala Jayanti ma'am. And I also would like to thank P.V. Reddy sir for permitting me to use beehives to conduct experiments. And I would like to thank Dr. Divij, Dr. Gandham Krishna, Dr. Meenal ma'am, and Dr. Jayanti Mala for their assisting me in several bioassays and technical aspects. And also thank my wife who's helping me in bioassays. I thank Mr. Madhusudan, who helped me initially in microbiological work, and his expertise in microbiology helped me to do all identification of the bacteria. And also I thank Dr. Arul providing the BioLivia software for doing molecular docking studies. And not but the least, I thank Dr. Vivek Kampraj for guiding some of this part of the experiments and guiding in my publications. And with this, I would like to conclude it. And thank you so much and giving me the opportunity to share my study with all of you guys. Thank you so much. Thank you, sir.
Thank you. Thank you so much, Sarwan, for your excellent presentation, particularly for accepting the invitation also, because only 2-3 days back I asked and you came forward. Yes, thank you so much. And very, very valuable points in all the objectives. Some of the application is there, how to use this these findings in application in the management of the this mark, particularly in honeybee industry. Here we
Yes,
have Dr. P.R. Reddy also, who himself is working on honeybees, and Dr.
Of
Uma
course.
Maheshwari, who is using this for nematode studies also. Now the topic is open for discussion.
Thank you, sir. Thank you so much.
Yeah, Shravan.
Yes, sir. Good afternoon, sir.
Yeah, good afternoon. Excellent presentation. And
Thank
you excuse me, actually, in between, I went out somewhere for work, so I missed some 5-10 minutes.
No problem, sir.
So maybe you might have covered. That's why I'm asking. Maybe I don't know you covered or not. This very nice. You have taken very rarely this beekeeping pests are addressed, especially in South India. Maximum work is done in case of mellifera in North India.
Correct, sir.
So this is a good attempt and approach. You have combined biotechnology, chemical ecology, and other things. But I think my impression is you have used this Galleria as one tool. I think your objective is much wider, I think, by using understanding its wax degradation mechanism and what is the role of the biosymbionts and all. So you want this outcome may be used for still large scale pest management beyond wax moth management.
Yeah, it can be used, sir, because anyhow now it is confirmed that this wax moth is responsible for this wax degradation and even the polythene degradation.
Yeah.
So if you're able to identify those kind of enzymes or those kind of genes which are responsible for that, you can use this information idea towards biotechnological approach, a molecular approach towards it, and you can use any other ways also, like not only as a pest management, you can use in other industries also, sir.
Other where the wax is used. Yeah. Okay. Then second thing, could you rear wax moth in lab?
Sir, actually we used to get it from GKVK, and whatever the available culture was there, we used to take it and provide with the honeycomb. Like we used to get from your field only, sir. Thanks to you for that.
Okay. Okay, nice. All the best.
Thank you, sir. Thank you.
Okay, next. Anybody else?
Yeah, some students should ask some question.
Yeah. Yes. Actually, he has shown so many means of expressing results. So many things he has shown. For those who are writing thesis, they will get maximum benefit from this.
Even slide making is very good.
Yes. Yeah.
I think somebody is messaging, sir.
Yeah. Some of the messages also you can see just in Kerala. Yeah, you can go through. You can go through yourself and respond. Yeah.
Okay, sir. Okay. Dr. Shivakumar, excellent work. Looking forward to field application of the studies. Thank you, sir. In Kerala, many people step back for apiculture due to wax moth attack. It is a serious problem in our area. Yeah, of course, sir. It is also a serious problem, not in your area. It's everywhere. If the proper sanitation is not been taken, the sanitation is taken, and if your beehive is healthy with a strong honeybee colonies, then you might not facing this problem. But anywhere, if you find a little, not putting your whole efforts in maintaining the sanitization, then it's gone case, sir. Wax moth will affect and it will attack it. It's like that.
Yeah. To comprehend your work for the management of this wax moth, what you suggest?
Sir, actually now farmers are becoming very intelligent. So as I shown in my slide, they only themselves coming with a different type of trap designing, and they're using it to prevent the wax moth. But as of suggestion you have asked me, very simple and very cheap method is proper sanitation. If the sanitization there will be no problem with the wax moth. But once the wax moth effect started, it's better to remove all this honeycomb the beehives wherever it is attacked and replace it with again a proper one. Whole the area should be eradicated because other control methods anyhow is there, like the chemical method, the temperature method, hot and the cold temperature, keeping all this existing control methods are there, but still it is not, it is limited, sir. It is not having that great potential towards it.
Okay, right. Anybody else? Everybody is satisfied with your explanations and presentation. All the best for your thesis presentation and your new assignment also. Brief thing about your new assignment, where you would like, where you will be going and joining.
Sir, as I said, actually I have got a good offer from USA, and the offer much belongs to the same work what I'm doing here. So it's almost about the chemical ecology lab, which I have to do some GCID and GCID studies. So the company is called Apex Biotechnology, and it is situated in California. So the documentation work has been going on, sir. So maybe a couple of months I'll be leaving, I think. Yeah.
Right. So congratulations for that. And last call, anybody has any clarifications? Otherwise, thank you once again from the association side. We profusely thank you, Saravanan, for coming forward, for giving sharing your experiences as well as your PhD work also. Thank you so much. With this, we'll be ending today's session, and we'll meet in the next month, which will be the first month of 2023, with a new speaker and a new topic. Thank you so much. Thank you, everybody, for joining.
Ends with my talk, sir, then.
Which one?
This year ends with my talk.
Yes.
It's my pleasure, sir.
Happily ending our series for this year. Thank you so much.
Thank you, sir. Thank you.
Okay, thank you. Thank you. Thank you, everybody. I'm closing this link.